Hello, my name is Carol Leibel, and today we want to examine cellular reproduction, or mitosis and cytokinesis, that occurs in eukaryotic cells. Now, technically, mitosis is the process by which a cell separates the chromosomes in its cell nucleus into two identical sets, and then will form two separate nuclei, or go through the process of nuclear division. It's generally followed immediately by cytokinesis, which divides the nuclei, the cytoplasm, organelles, and cell membrane into two cells containing roughly equal shares of these cellular components. Mitosis and cytokinesis together define the mitotic phase of the cell cycle, which is the division of the mother cell into two genetically identical daughter cells. M, or mitosis, accounts for about 10% of the cell cycle. Remember, the cell cycle, the cell will go through G1, S, and G2. And then this right up here represents mitosis. So there are two main purposes for cell reproduction. If you're a unicellular organism, going through mitosis will allow you to produce asexually two daughter cells. So that's perhaps an amoeba going through the process of mitosis. And then here are two yeast cells going through the process of mitosis. If you are a multicellular organism, you're going to do mitosis for cell division for growth or repair, or to make a new organism from a fertilized egg or sperm cell. It is indeed possible to have mitosis without cytokinesis, and this occurs quite often in fungal cells. This little diagram right here shows a hyphae in which we have mitosis occurring without cytokinesis, so what we have are multinucleated cells. The DNA in humans will be coiled into 46 chromosomes. It turns out that the number of chromosomes from species to species may vary. For example, humans have 46, chimpanzees have 48, elephants have 56, a lowly cabbage has 18, but a hedgehog has 90. The number of chromosomes really doesn't indicate the sophistication of the organism. And so, during the process of mitosis, the DNA has to be coiled into structures called chromosomes. And you may be asking yourself, why do you have to do that? Well, the reason you have to do that is if you can imagine a bowl of spaghetti and taking your hands and just driving them into the spaghetti, grabbing it and pulling very hard, a lot of the spaghetti strands are going to break. On the other hand, if you have small little compact spirals of pasta and you do the same thing, they're not going to break at all. And so the purpose of a chromosome is to prevent the DNA from breaking as it's being separated. We've mentioned this before. DNA is wrapped around histone proteins. Okay, so here's a histone protein. And then that's going to coil and coil upon itself to form a very small structure called a chromosome. And what's going to happen is that this chromosome, double-stranded chromosome, is going to separate into two single-stranded chromosomes during the process of mitosis. So let's look at chromatin. During interphase, DNA will form something called chromatin. And it turns out that there are areas of the DNA that are tightly wound around, that remain tightly wound around those proteins called histones. And then there are areas of DNA that are not wound or are loosely wound around those histones. The question is, what determines whether it's tightly wound or not tightly wound depends upon how the DNA is being used. Is it being transcribed for a particular gene, or is it being replicated? Chromatin is a complex in which we have about 40% DNA, and the rest is 60% protein. You also can find RNA associated with the chromatin if a particular area of the DNA is being transcribed. Heterochromatin is DNA that is not being expressed into proteins. As it turns out, about 90% of all your DNA is actually heterochromatin. Euchromatin is that part of the DNA that is going to be expressed into proteins or genes. When the DNA is being wrapped around these histones, there are about 200 nucleotides, and these histones actually form these complexes of 8 of those histones. And when the DNA is wrapped around 8 histones, it forms something called a nucleosome. And a nucleosome, underneath the electron microscope, kind of looks like a strand of pearls. Now, what's going to happen next in the formation of a chromosome is that those pearls are going to be coiled. Think about taking a string of pearls and wrapping them around a wooden dowel or a stick. When that happens, it's going to form a structure called a solenoid. Look at the next slide, and here we go. This here, right here, these are the solenoids. Okay? And now, when the chromosome, what's going to happen next is that's going to be looped around a scaffold. So if you could think about a pipe cleaner being wrapped around a stick, that is how we're going to form those chromosomes. Alright, so now, remember that the DNA has already been replicated. And so when the chromosomes are formed and they're double-stranded, because they have been through S, you're going to have 2 identical copies, and they're going to be held together at an area called the centromere. Okay? 1/2 of that double-stranded chromosome is called a chromatid. So what you're looking at right here is 1 chromosome with 2 chromatids. Now, depending upon the species, these chromatids may be actually kind of like sewed together or held together by some proteins called cohesions. Other species may only have the cohesions right here in the area of the centromere, like this photograph. So look at this picture right here, in which the cohesions are only present at the centromere, and look at this photograph right here. In this photograph, microphotograph, there are cohesions holding the chromatids together. This is a very nice graphic in which what you're looking at right here is the definition of chromatids versus chromosomes. So here is a chromosome. It has a single strand of DNA. So now, if that DNA has gone through the process of S and it's been replicated, you're going to have a double-stranded chromosome that's being held together by cohesions. And this is what it looks like after it's been through S. Now, after the process of mitosis, these chromatids are going to become single-stranded and chromosomes in their own right. So each daughter cell is going to get a copy of the chromosomes. Alright, so this graphic is illustrating the difference between something called a centromere and a kinetochore. A centromere is a visible region of constriction on the chromosome. It's like where it's being pinched in. The DNA found in the area of the centromere region consists of DNA repeats. Within the centromere region, there's a distinct disc-shaped structure made out of proteins called the kinetochore. So while that's the centromere region, that right there, that little disc-like shape, that's the kinetochore. It turns out that the kinetochore has an area in which microtubules can attach during the process of mitosis. Each chromatid has its own kinetochore. Now, I got to tell you that in many books, the terms centromere and kinetochore are used interchangeably, but technically, the centromere and the kinetochore are two different structures. And if you want to count the number of chromosomes in a cell, just simply count the number of centromeres. And so, in this little structure, you can see kinetochore spindle fibers attaching to the kinetochore. And these are two sister chromatids, and cohesions along the length of those chromatids have been broken down, and only cohesions at the centromere area are holding the two centromeres, the two chromatids together. So we're going to see a nice little introduction of the process of mitosis and cell division.
Mitosis, the division of the nucleus, has begun. Just before mitosis, the cell's centrosome duplicated itself, and now microtubules emanate from both centrosomes in all directions. Additionally, the nuclear membrane has broken down, and the chromosomes now occupy the center of the cell. Each chromosome consists of two identical sister chromatids attached together at a region called the centromere. At the centromere are disc-like anchorages called kinetochores. The kinetochores on the sister chromatids are captured by microtubules from opposite poles of the cell. At first, the chromosomes glide along the microtubules. The chromosomes oscillate between the two poles. This tug-of-war is orchestrated by an army of motor proteins and enzymes. Finally, the chromosomes are aligned between the two poles. Pulled by the microtubules connected to the poles, the chromosomes suddenly split apart. The function of mitosis, to separate the chromosomes into two identical sets, has been accomplished. Now, a contractile ring pinches the cell across its middle, forming two identical daughter cells. The two daughter cells will grow and divide in turn. This is the cell cycle, the basis of all life.
Alright, so mitosis has phases, if you will. You are not responsible for knowing the names of these phases. However, you are responsible for knowing what the sequence of the events. In terms of phases, you have prophase, prometaphase, metaphase, anaphase, and telophase. And we're going to go through each one of those, and we are going to tell you the names as we go through the sequence of events, just so you're aware of that. Okay? So the first thing that happens during the process of prophase is that the chromatin fibers begin to condense into chromosomes, and we begin to see them we can see them underneath the microscope. Cohesions are going to hold the chromatin arms together. However, we've already pointed out that if you're talking about a vertebrate, the cohesions are only going to be found in the centromere area. The nucleoli disappears in the nucleus, mitotic spindle fibers form asters radiating out from the centrosome. And after replicating, the centrosomes are going to start moving towards opposite poles. Now, centrosomes are microtubule organizing centers. These centers organize the microtubules to either attach the kinetochore or to overlap one another to help push the poles apart during the process of mitosis. So, and it also turns out that if you're talking about an animal cell or certain protista, or lower plant cells, you can also see another structure within the centrosome called a centriole. When you were talking about cell organelles, you'll remember that centrioles are composed of 9 triplets of microtubules, and that there's actually a pair of them and they're at right angles to one another. Now, if you take out the little centrioles, mitosis can still take place. It also seems to be important to help with the process of flagella and cilia formation. So one more time, eukaryotic cells have centrosomes. However, animal cells, lower plant cells, and certain fungi within those centrosoles have little structures called centrioles that are composed of microtubules. The function of the centrosome seems to be microtubule organizing centers. So what you can see in the process of prophase is that you can see that these the centrosomes have replicated, and now they're starting to move towards opposite poles. So let's look at this little animation about prophase.
During prophase, the nucleoli disappear and chromatin fibers coil up to become discrete chromosomes. Each chromosome consists of 2 identical sister chromatids, joined at the centromere. Microtubules grow out from the centrosomes, initiating formation of the mitotic spindle.
So now we want to talk about the second phase called prometaphase, and in many older textbooks you will not find this phase prometaphase. However, it's different from prophase and metaphase that they have given it its own name. So during prometaphase, the nuclear envelope fragments and the nucleolus is no longer visible. However, in many fungi and protists, the nuclear membrane remains intact and the microtubules will penetrate the nuclear membrane. If the nuclear membrane does not break down, it's called closed mitosis. But if the nuclear membrane fragments and breaks down, then it's called open mitosis. Of course, humans, not being fungi, we have open mitosis. In this particular diagram, you'll notice that here the chromatids are remaining together, and that's because of cohesions. Remember, it's only vertebrates in which that cohesion is no longer found the length of the chromatids, and so those little chromatids are just kind of moving back and forth. Alright, so the centrosomes are now at opposite ends of the nuclear area, and we see microtubules are extending through the nuclear area. 2 opposing kinetochores form on the centromere on each chromatids. Kinetochore microtubules are going to attach to the kinetochores, moving the chromosomes back and forth until they reach the middle. So here is the kinetochore, and we're going to find kinetochore microtubules attached to both sides, and now these guys are going to move back and forth until they move towards the metaphase plate. Non-kinetochore microtubules are going to overlap, and they're going to be important in the process of anaphase and separating the sister chromatids. So let's see this little animation about prometaphase.
During prometaphase, the nuclear envelope breaks into fragments. Some of the spindle fibers reach the chromosomes and attach to kinetochores, structures made of proteins and specific sections of DNA at the centromeres. Non-kinetochore microtubules overlap with those coming from the opposite pole.
This is just showing you a cell actually going through prometaphase. And here you can see radiating out from the nuclear area, these are non-kinetochore microtubules. And they're going to be used, as I said, in anaphase to push things apart. Alright, so metaphase is the longest phase of mitosis. And as at the end of metaphase, double-stranded chromosomes are going to line up on the metaphase plate. They have been tugging and pulling the microtubules, and finally they're lined up on the metaphase plate. So what I want you to do is to look at this and tell me how many chromosomes do you see and how many chromatids are there. So here's 1, 2, 3, 4, 5, 6 chromosomes. They are double-stranded. Each strand is a chromatid, so there are 12 chromatids. So now we're going to see a little animation on metaphase.
At metaphase, the mitotic spindle is fully formed, and the microtubules attached to kinetochores move the chromosomes to the metaphase plate, an imaginary plane equidistant from the poles.
Alright, so now we're going to look at anaphase. So during this next phase, cohesion proteins have to be cleaved so that the sister chromatids can be separated. Now, it turns out that this is a complicated process, and you don't have to memorize it, but just to let you know that there's a signal that's going to be transmitted called the anaphase promoting complex, or APC. The APC separates, deactivates something called the securin protein. Now, the securin protein was an inhibitor of an enzyme called protease, and a special kind of protease called separase. So once the securin has been deactivated and is no longer attached to separase, separase can do its duty, and it's activated, and it breaks down the cohesion proteins, holding the chromatids together. So now they can be separated. The other thing that the anaphase promoting complex does is that it will mark certain proteins for destruction by tagging them with that little protein known as ubiquitin. So looking at this particular picture here of anaphase, how many chromosomes do you see? And how many chromatids do you see? Well, now that these have become separated, they are chromosomes in their own right. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So there are 12 single-stranded chromosomes, and there are no chromatids because they've been separated. So how do these kinetochore microtubules work? You might be asking. So are they being pulled or are they being pushed? So let's look at an experiment that they did to show this. well, first let's look at this little animation with anaphase.
Anaphase begins when the two centromeres of each chromosome come apart, separating the sister chromatids. Once separate, each sister chromatid is considered a full-fledged daughter chromosome. Motor proteins of the kinetochores walk the daughter chromosomes along the spindle microtubules toward opposite poles, and the microtubules shorten. At the same time, the spindle microtubules not attached to chromosomes lengthen, pushing the two poles farther apart and elongating the cell.
So here's an actual movie of anaphase taking place. One more time. Alright, so the question is, which end of the microtubule is shortened? Is it this end or is it this end? I mean, you have an end here and an end here. So they did an experiment in which they were able to somehow mark these microtubules. And as they watched, it turns out that it is this end here that's being shortened. And so if you can imagine, the chromatids are kind of like walking up those microtubules, and as they're walking up those kinetochore microtubules, the microtubules are being destroyed, if you will. They're being disassembled. Alright, so telophase. What happens in telophase? In telophase, we now have the DNA separated, and we're going to have the formation of the nuclear membrane. So we're going to have two daughter nuclei within the cell. So the nuclear embryo envelope is forming. The chromosomes are going to form chromatin and disassemble, and we're going to see the beginning of cytokinesis. So here's a nice little animation showing that.
During telophase, nuclear envelopes form around the identical sets of chromosomes at the two poles of the cell. The chromosomes uncoil and nucleoli appear in the two new nuclei. Meanwhile, cytokinesis begins, splitting the cytoplasm and separating the two daughter cells.
So, as I have mentioned before, you can have mitosis without cytokinesis, and if that should happen, that's going to result in multinucleated cells. This happens in certain algae, fungi, and even a few animals. So when it comes time for cytokinesis to occur, animal cells do it by pinching in the cell membrane. Rings of a protein called actin forms underneath the cell membrane associated with myosin, and it pulls the membrane in kind of like a purse string and forming something called a cleavage furrow. So here you would have circular circular actin, and then you would have these radiating out here of myosin, and it's going to pinch in, forming something called a cleavage furrow. Hopefully it should separate most of the organelles evenly and produce two genetically identical daughter cells. So here's a little cytokinesis in algae. In algal cells, cytokinesis occurs by an inward growth of new wall and cell membrane. So here is the cell membrane is moving in the cell wall is moving in that direction and in that direction. However, in higher plant cells, you get this little patchwork of cell walls being put down and growing towards one another instead of just the ends moving inward. Okay? So here's one more time comparing cytokinesis in plants and animal cells. You get this cleavage furrow, contractile ring of microfilaments, and then you cleave the membrane in half and you get two daughter cells. But in plant cells, you get little vesicles forming and the cell wall being laid down and finally fusing, and then you separate the two cells in the cell plate formation. So here is a little movie showing you cytokinesis in plant cells versus animal cells.
The cell wall can be seen forming as many vesicles fuse along the center of the cell. Two new animal cells are formed as the cytoplasm pinches off between the nuclei.
Alright, so it shouldn't be surprising that we find mitosis occurring in plant cells mainly at the roots or at the tip of a stem or the plant. Mitosis doesn't occur anywhere in the plant. There are zones of areas where mitotic divisions take place. So here's a nice little overview of mitosis one more time.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. Two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
All right, so the very last thing we want to look at is the evolution of mitosis. So this is prokaryotic division by binary fission, and we've mentioned this when we talked about how the DNA is being replicated. The DNA, the circular DNA, attaches to the plasma membrane, and then we have the replication of the DNA forming two circles or two circular chromosomes, and then what will happen is that the cell wall and the plasma membrane will pinch in, and you will have two daughter cells that are genetically identical to the original cell. Okay, so it's much simpler. Well, the question is, how do you go from this to this very complicated, orchestrated process of mitosis? Well, here's some interesting things. Looking at yeast cells and diatoms are algae found out in the ocean, they do not break down their nuclear membrane. Instead, it pinches in very much like the binary fission of prokaryotic cells. The microtubules are contained within the nuclear membrane. So that's kind of interesting. And then from there, we move on to dinoflagellates. Now, dinoflagellates are also algal cells found out in the ocean, and they're a little bit different in that they do not break down their nuclear membrane, but the spindle fibers penetrate the nuclear membrane and attach to the plasma membrane. So like a little more sophisticated. Okay? And then finally, if you can imagine going from there to the process of mitosis, in which the nuclear membrane is totally broken down, and then mitosis with spindle fibers take place. So those are the steps that they believe happened in the process of going from binary fission to mitosis. Alright, so that's it, and we will talk to you later. And hope that you have a great day, and take care. Bye!